Hello, everyone, and welcome to ACS Webinars, connecting you with the best and brightest minds in chemistry, live from Washington, D.C. I'm Michael David, and I'll be your host for our last webinar of 2020, which is being co-produced with CAS, a specialist in scientific information solutions. In 1 year's time, the novel coronavirus has infected over 72 million people and has taken the lives of over 1.5 million worldwide. However, with the coming of a new year, there is new optimism as the scientific community has developed various potential vaccine candidates to address the global pandemic. Today, we will be joined by a panel of CAS experts to give us an update of those candidates, as well as the global perspective on the published research regarding COVID-19 vaccines. We would love to get you involved in the discussion, and you can share your thoughts and questions in the questions panel. You can also join the discussion on Twitter by including @AmeriChemSociety in your tweet. The slides for today's webinar can be accessed right now by clicking on the link that you see in the GoToWebinar control panel. And an invitation email to view the unedited recording from today's presentation will be sent to all of you once this broadcast is complete. Today's moderator is Jill Georges, who is the Vice President of Content Operation at CAS, as well as the Chief Scientific Officer. Jill will be leading today's presentation discussion and asking some questions throughout the presentation to make it more interactive. And with that, Jill, I will turn it over to you to get things started and introduce today's topic and speakers.
Thank you, Michael. Thank you very much. Hello, everyone. This is Jill Georges from CAS. Very happy to be with you today. So as an introduction, I want to say that ACS and CAS have been very engaged in the global research and development efforts to fight COVID-19. We put all the data we had available in our content database, and we offered it to anyone who could leverage it to accelerate the discovery for a solution. We offered data sets, documents, high quality papers, anything that could have value. And since the breakout of the COVID-19 pandemic, we've been closely monitoring the status of R&D efforts for therapeutics, diagnostics, but also vaccines. And as you know now, after the announcement of the vaccine approvals in the US and also in Europe, probably a good time for us to look at those solutions and see what they can offer and what they mean to us. So I'm in very good company today. As Michael said, I have 3 bright scientists with me to cover the topic and provide you with an update that will hopefully enlighten you on those vaccines, how they work, the immunity they may provide, and the impact they potentially could have on the pandemic and our health in the future. So I have with me Dr. Angela Zhu, Dr. Cynthia Liu, and Dr. Catherine Safeson. Welcome, ladies, and thank you for being with me today. I would like this to be a dialogue with you, and as much as possible, I have some questions for you, and I know that you have those answers somewhere in your session here today. And at the end of this, as Michael said, we have a Q&A from our listeners today. But I want to start with you, Angela. And before we go too much into the vaccine technical details, I think most of us would like to understand how a vaccine prepares our immune system to fight against a disease. And could you please share some light on the protection mechanisms that the vaccine triggers in our body?
Sure, Jill. You actually asked me the exact right question for me to begin with. Now, before we move into the details of COVID-19 vaccines, I do wanted to share a little bit of the background information with you of how our immune systems can be trained by a vaccine to help us prepare for future viral infections. So as we know that most of the vaccines will start with a needle injection, that injection will bring in a solution to our body. That solution often contains called antigen. So these antigens could be live attenuated virus, sometimes it could be killed virus, or sometimes could be just, parts of the viral proteins. These antigen proteins, once they get into our body, they can actually interact with the cells of our body. And there are a particular group of cell, we call them APC cell, or sometimes we call them antigen presenting cells. As you can see from this picture that those cells have long protrusions. So it is very easy for these cells to engulf anything surrounding them. So they can intake these antigen proteins within the cell body and then process the information. these cells, when they engulf these antigen proteins, they may see, this is something new that I've never encountered before. So then they will process the information and display at the surface of this APC cell as invader signals. So once they display these invader signals can actually train our immune cells, such as B cell and a T cell, train them, help them to produce the specific antibody, for example, from the B cell, to produce those specific antibody that these antibody can bind to those antigens. So in the future, if there are virus invaded, that particular virus, invaded in our body, these antibodies will be able to bind to those viruses and neutralize those viruses. Now we know that these antibodies can only work for the virus outside of the cell. When the virus gets into the cell, it becomes a little bit tricky. That will require another type of immunity. We call those cytotoxic T cells. These cytotoxic T cells can be activated by the effector T cells, and we also call these cytotoxic T cells like killer cells. So the killer T cells can actually kill the cells that has been infected by that particular virus. So can kill those cells before the virus gets spread, out of the cell. So that is how our immune system can be trained, by a particular vaccine and get the system to produce these specific immunity. So when the real virus invades, we can, get ready to fight those viruses. Does that make sense to you?
Yeah, it makes sense. So let me summarize this in my own simple terms. So if the immune cells, as you said, are the guardians of our body, then the antigen will be the kind of dummy enemy to train our guardian cells, right? Is that what
Exactly.
it is? Okay,
Yes.
so when the real enemy invade our body, those immune cells can quickly recognize the threat and attack it, basically. That's how it works.
Yes, exactly.
Okay.
So being trained ahead of time, it actually saves some time for our immune system to respond before the virus replicate too much to cause any damage.
Okay. So what about that vaccine-induced immunity you talked about? How long is that supposed to last?
Great question. Yeah. So we know that these antibodies produced, it may decline quickly, like within a couple of months. And we have also seen that in the recovered COVID-19 patients, that their neutralizing antibody may start to decline within, a couple of months. However, the good news is that we have also been seeing those in vaccinated people or the recovered COVID-19 patient, that in addition to these antibodies, in our immune system, we also produce another group of immune cells. We call those mammary immune cells. Specifically, we call those mammary B cell or mammary T cells. These mammary cells, they can last for at least 6 months, and sometimes they may last for years. And these memory cells, they are able to remember how these invader signals look like for a long time, for a couple of years, 6 months to a couple of years. So that actually help us, to remain this immune memory in our body. In the future, if there's any these exact virus invades, our immune system will be able to respond quickly.
Got it, Angela. And yesterday we just heard about this new mutant coronavirus strain that shut down the UK. So could this new strain compromise the efficacy of those vaccines or those approved vaccines we just heard about?
Yeah, great question. So that is the concern. People are wondering, the immunity may last long. We are claiming it's likely going to last for at least 6 months to a couple of years. But what about the virus mutates, within the next couple of years? Will the vaccine still be effective to cope with this virus? From what I know, that most of the vaccines currently being developed against COVID-19, they're using the full length of spike protein as antigen. So we're really looking at is a complete, protein of the spike protein. That spike protein is about 1,700 amino acids long. And from the mutated strain, we can tell there may be 8 to 10, mutated amino acids. I would say, 10 out of 1,700, that's a relatively small number. The antibody actually produced will recognize the, the whole spike protein. So most of the experts are not worried so much of these, that virus escaping from the current vaccines. But we will see. Yeah. So
thank you, Angela.
yeah, sure. I think we've also heard a lot from news these days about different types of vaccine, how they are being developed,
Right.
and people may wondering, what's the difference between each types of vaccine and what's the advantage and disadvantage. So here I'm going to give you a very high-level overview of how these each vaccine strategies evolved. Yeah. So as you can see from the slides that I'm showing you, two vaccine platforms. One is called live attenuated virus vaccine. The other is called inactivated virus vaccine. So these two platforms are relatively early type of platform. In the live attenuated virus platform, people use those modified virus. That virus, they can still replicate, which means they're still live virus, and they can replicate in our body and stimulate our immune responses. However, this has been attenuated, which means they can no longer cause any disease. So it's safe. We do have existing, vaccines using these platforms. For example, the BCG vaccine or the measles vaccine, has been around for a long, long time. And the measles vaccine actually was very successful. It almost eradicated the measles disease in the United States and wiped out, decreased significant disease incidence globally. Currently, there are a couple COVID-19 candidate vaccines being developed using this platform. The inactivated virus vaccine platform, instead, it uses the whole virus, which has been killed by a chemical, or sometimes being killed by heat or radiations. There's no live virus anymore, but their whole virus has been killed. So it carries in the overall, viral proteins, but the virus itself is no longer active. So we do have some existing vaccines being approved using this platform. For example, the rabies virus and the hepatitis A virus. Both are very good, actually, showing good efficacies. And currently, there are about 22 COVID candidate vaccines being developed using this strategy. And moving forward a little bit, here I'm showing you, this is called viral protein vaccine. In this strategy, people use a little bit advanced technology called recombinant protein. So instead of culturing live virus, like the other two platforms, in this viral protein vaccine platform, we actually use the technology to synthesize protein in vitro, and then use this protein as antigen to produce the vaccine. And currently there are HBV vaccine and a human papilloma virus vaccine, HPV, both are developed using this technology. And as you can see, in a COVID-19 candidate vaccines pipeline, there are a lot of candidate vaccines currently being developed using this technology. And even more recently, you probably have heard a lot of these two newer platforms.
Right.
One is called, viral vector vaccine, and the other is called mRNA vaccines. So these two strategies actually using very different concepts. Instead of delivering the antigen protein itself to our body, in these two newer platforms, people use genetic information. They deliver genetic information to our body and asking our cells to make those antigen proteins in the cells. So the concept is very different, than the traditional ways. And in this viral vector vaccine, we use another harmless virus to carry the genetic code that gives cells the instruction to make the antigenic protein in our body. And there is a current existing approved vaccine, Ebola vaccine, which is conditionally approved in Europe, using these, viral vector vaccine platform. And currently there are about 50 candidate vaccines being developed using these viral vector vaccine platforms. And even more, mRNA vaccines, even a newer technology. In this platform, instead of using DNA-related genetic codes, we use mRNA genetic information to tell our cells how to make the antigenic protein. So we all heard, these exciting news from Pfizer and Moderna, these two mRNA vaccines being recently conditionally approved in US as well as, some other countries. So this is a very interesting, there are a lot of advantages, if we would compare with the other vaccine platforms. Yeah.
Interesting, and again, that's a good point here, Angela. I mean, this mRNA vaccine technology seems to be very prominent. I mean, you just mentioned a couple of examples here, but it seems like a lot of other companies are also looking at this technology, but it's a very new technology, certainly very promising, but very new and never really implemented before. Could you elaborate why it's been so not so successful, but at least prioritized as an approach and how it works really?
Great question. Yeah. So this actually goes back to the beginning of pandemic. At very early of the pandemic, people are already looking for vaccines. If it's possible, we develop a vaccine to cope, to fight against this pandemic. And mRNA vaccine really has been attracted with a lot of hopes and promises. Two things I could say here. One is the speed. With this type of approach, we can quickly adapt the platform to any new type of viruses, as long as the antigen is clear. So in this case, with the coronavirus, they accumulated research about spike protein being the best, antigen. So once the Chinese scientists, they released the genetic information about SARS-CoV-2 virus, people equipped with the mRNA vaccine platforms, they can quickly dive in, just use these genetic information will be enough for them to adapt the system to this new virus. And that's one reason. And the second reason is about, in this pandemic, we're really looking for big or huge productions, to help people, that every person on the earth can get vaccinated. And using this technology, we know that there's no need to culture live viruses. So it's much easier to handle in a manufactured perspective. And there's also no need for us to kind of optimize how to purify proteins like the viral protein vaccine will need to do. So here, virtually all the manufacture process will be the same, except just switching the code.
Yeah. Got you. So how does it work really?
Great question. So we do have some slides prepared. I'm gonna actually introduce my colleague, Catherine here, to tell you more about, these mRNA vaccines, how do they work.
Thank you, Angela. Catherine, welcome to you.
Thank you. Before I dive into the details of mRNA vaccines, I want to remind you of a basic concept that you may remember from your biology classes in school, that in human cells, genetic information is encoded in DNA. That information is copied into messenger RNAs or mRNAs, from which are made individual proteins. An mRNA vaccine delivers synthetic messenger RNA into a human cell. That mRNA encodes a protein antigen. So for COVID-19 vaccine, the antigen that we're interested in is the spike protein. Spike protein forms those knobs you see on the outside of the virus capsule. That's the part of the virus that interacts with a human cell during infection. To produce the vaccine, the genetic information encoding spike protein is used to produce synthetic mRNAs in the laboratory. That synthetic mRNA has all the components of a normal human messenger RNA that the cell machinery needs to recognize it, attach to it, and make proteins from it. So next, please. The synthetic mRNA is then packaged into a lipid nanoparticle for delivery to human cells. Next. So when the vaccine containing those lipid nanoparticles is injected into a human, the nanoparticles fuse with the antigen presenting cells, or APC cells, and the mRNA is released into the cytoplasm of the cells. So remember that those mRNAs, they look like normal human mRNAs. So the cell machinery attaches and ribosomes start making viral spike proteins from those synthetic mRNAs inside human cells. I want to mention that we recently produced an animated video to show how this works inside the cell. The video is on the CAS YouTube channel, and we'll put the link in the chat at the end of this presentation. Next slide. So once the spike protein is made, it's used to activate immune cells, just like Angela described. Next slide. And then antibodies specific to the spike protein are produced. So Gilles, the goal of this vaccine is to train your immune system against that spike protein. That's the antigen. So that in the future, when you encounter a coronavirus all covered in spike proteins, your immune system recognizes it and is ready to protect you from it. Does that make sense?
Yeah, that's fascinating, really, Catherine. It's incredible to see that. We heard in the news and scientific reports that those vaccines need to be stored at a very low temperature. Can you explain why?
Sure. So mRNA is fragile, it's inherently unstable, and it's prone to degradation. So that ultra cold storage temperature is necessary to keep it from degrading.
Okay, pretty straightforward. Another question for you, Catherine, and I've got to apologize for this one. May seem a little odd here, but this mRNA vaccine carries the virus genetic information in our body. So is there a risk that this genetic code could blend with our human genome?
So great question. The short answer is no, for several reasons. So
okay, feel better.
Good. The mRNA never enters the nucleus of the cell, and the nucleus is where our genomic DNA is. Also, our cells don't have the necessary machinery to integrate that messenger RNA into our genome. Also, remember what I said about stability. mRNA is inherently unstable. There are a lot of enzymes in the cell that will degrade it. So it's broken down by the cell's normal metabolic processes, and it doesn't stick around long term. Does that help?
That helps. Thank you very much, Catherine. Very good explanation. I think you have some information about the vaccine candidates right now that are being proposed to the public. And yeah, thank you very much. Please go ahead.
I do, thanks. So I'd like to introduce 3 important vaccines that have either been approved or are getting close. The companies involved are Pfizer in partnership with BioNTech, Moderna, and AstraZeneca in partnership with Oxford University. Pfizer and Moderna both have been granted emergency use authorization by the US FDA. AstraZeneca phase 3 trials were published earlier this month, and they are getting close as well. So as we mentioned, Pfizer and Moderna are both mRNA vaccines. AstraZeneca vaccine is based on an adenovirus vector, which is a weakened version of a chimpanzee common cold virus that can't grow in humans. So I'd like to call attention to the efficacy numbers here. I imagine that many of you got a flu shot this fall, right? I know I got mine in September. The average efficacy of a flu shot is 40 to 60% in any given year, depending on what strains are circulating. So the fact that these are all showing above 70% efficacy is remarkable, and it's really good news.
Yeah, and that's really striking me here, Catherine. So can you explain a little more how they achieve such a high efficacy percentage and also the speed of development? It's just quite amazing. Can you comment on this a little bit?
Oh, sure. So some reasons that may contribute to the high efficacy include coronavirus changes more slowly than the flu virus, so it doesn't mutate as quickly, it doesn't mutate in the same way. Also, these newer vaccine platforms synthesize the viral proteins right inside the human cells, so the proteins aren't being transported and stored. And the efficacy data is only based on clinical trials so far. I think it will be very interesting to see if those numbers change as more data becomes available. And then you asked about how these vaccines were able to be developed so fast.
So quickly, yes, exactly.
So there has been unprecedented collaboration and sharing of information among researchers around the globe. As Angela mentioned, Chinese scientists released the genomic sequence of the SARS-CoV-2 virus in early January. That was really early in the pandemic. There was a lot of financial and regulatory support from global governments. For example, Operation Warp Speed here in the US distributed billions of dollars for research and development upfront. And finally, because we're in the middle of a pandemic, there's a lot of public interest, right? People are paying attention, and there have been willing participants for these clinical trials.
Excellent, thank you.
Sure.
That helps.
Okay, so maybe we can move to the next slide. So to achieve that high efficacy, all 3 of these vaccines require 2 doses spaced either 3 or 4 weeks apart. And all 3 have been or are being tested in tens of thousands of volunteers. Next slide. All 3 of these companies plan to produce billions of doses in 2021. Pfizer and Moderna already have millions of doses available, and they're being distributed right now. And we already touched on the cold storage requirements. So mRNA vaccines, in particular that Pfizer vaccine, require super cold storage and transport conditions. That requires a cold chain of storage and transport companies that is going to be challenging and crucial for their effective distribution. The AstraZeneca vaccine can be stored at refrigerator temperatures, which makes its storage and distribution a bit easier. Does that make sense, Jill?
Yes, that makes perfect sense, Catherine. Thank you very much. This is a great slide. And in fact, I think we have now a survey. And Michael, I'm going to turn it back over to you for the survey.
All right. Our question for all of you is, how many COVID-19 candidate vaccines are currently in clinical trials worldwide? And you can answer directly just on this blue screen in front of you by clicking either only a handful, around 10 to 15, around 50, 100, or a few hundred. And I'm seeing the results start to come in. And we'll give you just a few seconds more to respond. When I close this in 5, 4, 3, 2, 1. And it looks like, I'll just run through them all since they all had pretty good responses. 36% said a few hundred, 17% said 100, 28% said around 50, 12% said around 10 to 15, and 7% said only a handful. And with that, I'll turn it back over to you.
Yeah, thank you, Michael. And we'll give you the right answer for this, okay? So bear with us a little bit. But before we do that, I want to bring in Cynthia Liu, Dr. Cynthia Liu. Cynthia is one of our biologic experts for content operations, and she will help us understand how we leverage our content, CS content, to help the progress on vaccines for the COVID-19. Cynthia, back to you.
Okay, thank you, Joe. And so here I'm just going to show a little bit of a data analysis we did recently. Here we asked the question where the COVID-19 vaccines are currently being developed around the world. And I'll try to show you the answer here. You can see that already. As of December 8th, there were 214 COVID-19 vaccines in the development pipeline around the world. And among these, 52 were in clinical trials. Actually, as of today, these numbers have increased to 222 and 56 respectively. So the correct answer is around 50. And indeed, as you all know, that two vaccines have already been authorized for emergency use. And the chart here though shows the top 15 countries out of about 40 countries in terms of number of vaccines being developed. there. And the orange color is for preclinical studies and blue color is for clinical trials.
Is it,
sure.
Sorry, is it possible? Just a quick one here. Is it possible that so a lot of countries have developed vaccines, and I'm assuming this is for primarily for their own population. But is it possible that a vaccine, for example, developed and approved in Turkey could also be approved here in the US, as an example?
That's an interesting question, Jill. I think vaccine developers typically develop vaccine for their own countries, but they also strive for taking their products to international markets. So if a Turkey-made vaccine is superior to a US-made vaccine in terms of efficacy, safety, cost, formulation, and so forth, theoretically, it can be approved in the US. Actually, in the case of Pfizer's mRNA vaccines, it was first developed by BioNTech in Germany. So that's an illustration of getting approved in another country.
Got it. Thanks, Cynthia.
You're welcome. So we then, as you can see here, the top 3 countries, US, China, and UK, in terms of number of vaccines being developed there. Then, can I have the next slide, please? Yeah, thank you. We then try to answer the question, what the COVID-19 vaccine research has focused on so far. We did that by analyzing published journal articles related to COVID-19 vaccines. And the chart here shows the distribution of those journal papers among different topics. And as expected, research paper focusing on the development of vaccine using various vaccine platforms or technology have the largest number of publications. But there are also significant number of publications that examine the epitopes, that is the regions in the viral antigens that can be detected by human antibodies. And there are also papers examining the mutations of this virus. We then determined which vaccine platform or technology is being explored more than others. So this data is shown on the right here. And as you can see, the protein subunit based vaccine platform or technology indeed got the largest number of publications, which is then followed by viral vector-based vaccine and the RNA or mRNA-based vaccine. So even though there has been a lot of news about mRNA vaccines, the protein subunit-based vaccine has also got a lot of attention. And so, Jill, as you know, there has been a remarkable progress as highlighted by the approval of 2 vaccines already after such short period of time. So we can start to be optimistic about the chances to get this pandemic under control. But going forward, there are still many challenges and unsolved questions. And here I only list a few. The first question is how long a COVID-19 vaccine-induced immunity will last and protect us from this disease. As Angela and you actually already alluded a little bit a moment ago. And I would say we don't know for sure yet because this is a new disease and the vaccine effect has only been monitored so far for several months. But it's likely that it will last beyond several months or even years. We'll see. And the second question is related to safety. That is, are these vaccines safe given the fact they were developed and given EUA status at an unprecedented speed? So while all the data that have been reported so far indicate that the authorized vaccines are as safe as other vaccines already in the market for years. And there is no cutting corners or any type of compromise in terms of safety standard in clinical trials or the review process for approval. And also the good thing is that there will be also post-market surveillance to continue to monitor the safety and efficacy of these vaccines. So we can feel good about it, feel comfortable, have more confidence.
That's important, very important,
Yeah.
Thank you.
And the third question would be related to herd immunity or population immunity. You know, how
And
so?
to that end, Cynthia, since
Sure.
you mentioned there's a herd immunity, what would be the percentage of population that would need to be vaccinated to generate this herd immunity?
That's a good question. Various numbers ranging from 60 to 85% have been proposed, but experts actually do not know the exact number yet. This is because a herd immunity varies from disease to disease. For example, in the case of measles, that number is actually 94%. And last question, we're all eager to go back to pre-pandemic life. And we want to know when we can resume our normal life.
Yeah.
Well, well, it's difficult to speculate at this moment, but because it depends on how effective these vaccines will eventually be, and whether we'll have a large enough percentage of population vaccinated in the next several months, and whether the viral transmissibility changes. And we already see one mutation that impacts the transmissibility, and whether everyone continues to practice social distancing and wear masks until enough people are vaccinated. So we probably won't be able to completely return to pre-pandemic lifestyles before next fall, even the end of next year. But hopefully we'll be able to resume some part of our normal life before then. Until then, though, we'll have to wear masks and practice social distancing. So I think that's all from me. I will now turn this back to you, Jill.
Thank you, Cynthia. This is a really safe reminder, and this is great advice here. And thank you, Cynthia. Thank you to Angela and also Catherine for their contribution here. And here on the slide, just the documents and material that I was talking about at the beginning here of this session, all the resources you can find on CS website, you know, the papers, the data sets, and also the video, the videos and the blogs, but the video that Catherine was talking about at the beginning about the mRNA vaccine and how that works. A very nice little video clip here that you can watch. So this is all this. Don't hesitate to visit us and see, find for yourself all this great information. I think it's time now to go for some questions. And I'm just going to browse over the questions that we received from you guys, and thank you for a lot of questions, a lot of them. So let me go through a few of them here. So first question is, how can you help with our relatives or friends to build confidence with taking the vaccine? Angela, do you want to take this one?
Sure, yeah, I would love to. As you know, as a scientist and as working in this field, since the pandemic, I do get these type of questions a lot from my friends and relatives. I think most of the concerns are coming from the safety, and people also have doubt about the speed, why this vaccine come out so fast. So I can touch on these two aspects. About safety, we know that most of the vaccines so far have been developed, approved, towards other diseases. Most of these severe adverse effects happens within 6 months after the injection, after the vaccine injection. So that is also the reason why FDA insisted at the beginning, we have to have 2 months worth of safety data for this vaccine to be approved as an EUA. So I think, adverse effect wise, it's relatively safe. We have, 2 months worth of data with large population. And also go beyond with the size of clinical trial with Pfizer and Moderna together, and there's AstraZeneca and other clinical trials here all together. We have several, we have tens of thousands of participants that have been tested with these vaccines, and the size is comparable with all the previous vaccines that has been developed. So that too, keep in mind, the FDA or the other authorities from other countries, we're not cutting any safety corners, to get these vaccines approved. Also, like Cynthia mentioned, there will be post-marketing surveillance systems. Anyone, if they receive side effects, severe side effect, they will report to that system immediately. We also seen there a couple allergic reactions, right, happen in the past, but those are very few. Maybe people, when they're receiving vaccines, they will continuously be monitored for at least half hour. That will be helpful to prevent severe consequences happen. And then theoretically, really, the mRNA vaccine is safe compared to live attenuated virus vaccine or we would call DNA vaccine, because mRNA molecule itself is very transient, like Catherine mentioned. It will degrade quickly. Maybe within days, if the molecule will disappear, and there's no way this molecule can integrate with our genomic sequences. I also got a lot of question about the speed, how we can rely on these vaccines, they being developed within 10 months, because usually vaccines will be developed, within like 10 years or, so that's a remarkable speed. I think these, it's not a quick turnover, because we do have a lot of research accumulated even before the pandemic. People have been studying the SARS and the MERS, when these two episodes happen. So we know that spike protein is the most ideal antigen. And also by knowing that the mRNA technology, it may sound new to us, but in reality, it's not that new. People have been researching on that technology for more than 10 years. So they know how to quickly adapt into a new virus, so that actually also helped the speed. And other, like Catherine mentioned, there are government support and available of, patients, willing to participate in the clinical trials. All these help to make the process much faster. And there are also a lot of financial risks that the world is willing to take at the beginning, right? We're actually manufacturing even before these get approved. Yeah.
Thank you, Angela. Great. Next question would be around the side effects. So what would be the most severe side effects that would be appearing after vaccination, potentially? Angela, you want to take that one?
Yeah, I can take that one as well.
Okay.
So the side effect, usually the side effect will happen within the first day or second day, after the vaccination. We call these side effects, they're not really side effects, they are expected. They expected immunoreactivity, which means your immune system is responding to the vaccine. They are working, they're practicing, right? It's almost like we go to gym and a workout, so you feel a little bit sore. They are practicing on these fake enemies. So that's how you got your immunity trained. But there are severe, you would call severe side effects. Usually those will happen, from previous vaccine development experience. Those always happen within 6 weeks after the injection. Yeah. So we have 2 months of safety data to follow. I think it's pretty solid.
Okay, thank you, Angela. And there is a question here. So there's a lot of questions around some groups of a population that potentially more at risk, and some of our listeners are interested about pregnant women, for example, the risk for pregnant women, but also the people that are immunosuppressed due to, for example, treatment to chemotherapy, and also the seniors. Would you have like a general comment to reassure us a bit on for those people that could be at risk in receiving the vaccine?
So I guess I can take that.
Or Catherine, yeah, thank you.
So it hasn't, the vaccines haven't been tested on pregnant women or lactating women. So the general advice is talk to your doctor, but that's normal common sense. For immunocompromised, also, we don't have data. So you would need to talk to a doctor and weigh the risk. So is the risk of contracting COVID and everything that goes with it higher than other risks, the risk of the vaccine? Let's see, in seniors, there is actually data on people over 65 for both the Pfizer and the Mrna vaccine. So they are as effective in older folks as they are in younger. So I would say the data is good for seniors. For other conditions, talk to your doctor.
Yeah, I think maybe there will be more data being released in the next few months, because for those participants, they were in the initial Pfizer or Moderna clinical trials. They actually become pregnant. You know, they realize they're pregnant later on. So we will have more data later on. And I also think CDC is recommending, if a person, I pay particular attention to pregnant people. If the person is pregnant and he or she is also, she is also in the recommended group, like if she is a healthcare provider, they recommend that person get the vaccine if the person is willing to. You know, of course, they need to consult their doctors as well. Yeah.
Thank you. I know it's difficult, question and answer probably, but that helps, I think. Thank you. Another question, I think, Cynthia, you alluded to this a little bit, but the question is, would the safety of the vaccine be continuously monitored after it has been given to the general population?
Yes, I alluded to that. Yes, absolutely, yes. And actually, both companies, Pfizer and Moderna, plan to continue to monitor these for at least 2 years. Yeah, so okay. we can feel confident on that.
Okay, thank you. The next question, guys, and there's a lot of them, so I'll try to really pick the ones that are a little different and relevant for as many people as possible. But the next one I would pick is, are the vaccine clinical trials participants diverse enough to represent the overall demography? Catherine, I think you can probably answer this one.
Yeah, so both Pfizer and Moderna made a concerted effort to get a diverse test population. So Pfizer expanded the size of its study in order to get enough non-white participants to be significant. You notice they had something like 44,000 participants in their clinical trial. Moderna extended the length of time of their tests, their clinical trials, in order to get enough participants. And AstraZeneca is testing in the UK and South Africa and Brazil, I think. So there's been a concerted effort to get good, diverse population data for all of these vaccines.
Thank you. Thank you very much. So next question. How does the vaccine-induced immunity compare to the natural infection immunity? Angela, can you take this one?
Sure, yeah, I would love to. So that's actually a very good question. You know, we do have people recovered from COVID-19, and they're wondering if they should take the vaccine, if taking the vaccine will be beneficial to them. We don't have much solid data yet, because it takes time for people to accumulate data. But overall, just from a scientist's perspective, okay, I think the vaccine-produced immunity may be stronger than the natural infections. There are several reasons. First, because the COVID, virus, the SARS-CoV-2, has some kind of mechanism that will inhibit our immune response, interfere our immune response, and that is why we are seeing a lot of, in severe cases. So the immunity produced by the, natural infection may be some kind of inhibited by the viral mechanism, And when we introduce in vaccine, we produce that spike protein without any other, immuno-interfering mechanisms. So it's more pure of immune provoking mechanism. So I think theoretically it will be stronger. And also we are concentrating the antigens. So just the spike protein, right? And in a natural infection, there will be a broader range of antigens present, and you may have antibodies towards different types of antigens on the virus, which is good. But another way, because the spike protein is really the receptor binding to our cells, so by blocking that spike protein with our antibody is actually very potent, and we've been showing that a lot of neutralizing antibodies actually they are against the spike protein. Not much of a neutralizing antibody has been discovered, against other parts of the virus yet. And the good news is that, yeah, Moderna actually tested the vaccinated individuals, and they're showing their antibody lasts for at least 3 months. And there are also historical vaccines we know, like tetanus vaccine. They actually produce stronger immunity than natural infection. So there's a reason to believe that, yeah.
Thank you, Angela. And there's a question I think that a lot of parents will be interested by this question. Any information on vaccines either in development or in trial for children? Maybe Catherine? Yeah.
Yeah, sure, I can take that one. So there's some data from Pfizer on children ages 12 and up. I think those trials started in October, maybe. Moderna's starting trials on children 12 and up this month. So there's not enough data yet to say anything definitively, but Pfizer was approved by the US FDA for ages 16 and up. So they haven't gotten to testing younger children yet, but they'll work their way back, I think.
Okay. And there's a question related to that. Why weren't children included in clinical trials for the vaccine on this first wave?
Well, they start with adults first. Anybody else have a thought?
Yeah, I can take, answer part of the question, I guess. Children, their immune response is much stronger than adults. The dynamics are very different. So we have to be really careful to test those in kids. The younger they are, their immunity could be more stronger or very different than adult. So it's always safe to, if this is a disease happening in all age population, it's always safe to test in adult first, make sure it's safe, then move on to the next stage with kids.
Got it. Thank you, Angela and Catherine. We have also a lot of very technical questions, so I want to touch on that a little bit for the people also interested in the science behind those vaccines. So this one is, any thoughts on why the Oxford vaccine elicits a better T cell response versus a better B cell response for the mRNA vaccines? Angela, you want to take this one?
Sure, yeah. I honestly, actually, I don't know why. I can only guess. But for us to elicit these immune responses, there are two mechanisms. One is to activate through the antigen presenting process. One is through MHC1, the other is MHC2, and the one of them, they can actually activate the helper T cells, and that helper T cells can activate the effector T cells and etc. I'm not sure. Actually, I don't know why you're saying AstraZeneca can produce stronger cell-mediated immune response. It could be they're mimicking more as the viral infection, because the antigens will be presented on the by the APC cells, more like the natural infection, and you may have less secreted proteins on the surrounding environment. But I don't know. I also don't know how to compare with different vaccine strategies, because they may be measuring these responses differently, using different laboratory methods, to measure the antibody titers or the frequencies of memory cells. Yeah, that all I can share about my insights here.
Thank you, Angela. Thank you. Another one that's also quite technical. Can you make recombinant spike protein and use that as a vaccine? Angela,
of course. Of course.
Okay.
They are actually, right now seems the front runners are all the mRNA vaccines or the viral vector vaccines, but there are other vaccines. They are being developed relatively slower, taking slower pace. but they may be able to overcome a lot of shortcomings that these front runners, cannot. So there are protein-based vaccines. They actually use the spike protein as the antigen. So they actually synthesize the whole spike protein in vitro as a antigen, and then can use that as vaccine to, induce our immune response. So there are a couple of very, very promising protein-based vaccines currently being developed. And these types of vaccine actually, the public will have higher confidence on those because there are existing, approved vaccines using these platforms. And also they may overcome these, logistic challenges of temperature requirements and so on. It's just their manufacturing process may take a little bit longer because they have to purify the protein, and the formulation is a little bit more complicated. The FDA may take a longer time to approve that.
Thanks, Angela. And any idea why the COVID-19 virus doesn't mutate as quickly as other viruses? Is that a common trait of coronaviruses?
I can take that question as well, and Cynthia and Catherine, if you can add, please feel free. The coronavirus itself is a relatively sophisticated virus if we were to compare with flu virus. If you look at the genomic information of these viruses actually, other than the RDRP, which is the RNA-dependent RNA polymerase, which is to replicate its genomic information, other than that enzymatic activity, these viruses also has a proofreading functions. Almost like our eukaryotic cells, we have proofreading functions, make sure the replication goes correctly. So these viruses has a proofreading functions. That's why you see fewer mutations, as compared to other viruses. And also virus mutates when they take a longer time incubating in our body. I think most patients may have these virus infected and they may be able to get rid of the virus within a couple of weeks. So only have a short period of time. Only in those patients, they may have positive viral infection for a couple of months. For a longer period of time incubation, then they may accumulate more time for them to mutate. Yeah, so all these factors we can consider together. Yeah.
Thank you, Angela. And we have, I think, time for one more. And if you can give us a quick answer on this one, it's kind of the $1 million question here. If you had all type of vaccines available, which one would you choose? So if they're all available, is there a preference or is there a choice we should go to first, or should we wait to see what are the ones that are really efficient before, or have a good efficacy before we make a choice? What is your perspective on this, Angela, Cynthia, and Catherine? As quickly as possible.
Maybe I'll say my choice first, and then we can all have different choice. Based on the efficacy of the mRNA vaccines, and so far the side effect, plus the urgency. And so we don't want to wait for too long, and we want to resume to normal life as soon as possible. So I may go with mRNA vaccine.
Okay.
Others may have different feeling.
Yes. All right, guys. Hey, we're very close to the end of this session, and there's a lot of other great questions, and apologies for our listeners. We cannot cover all of them. They're very, very good questions, but hopefully we were able to give you a little more information about, the vaccines, how they work, and how they could potentially protect us. And there's still a lot of, time for us to see the efficacy of those vaccines. But I want to thank you, everyone, the participants, Cynthia, Catherine, and Angela, for covering all this incredible good topic today. It was really great, and it helped everyone, I'm sure. So if I could ask a last question here, if there's one thing that we would like our listeners to remember from today's webinar, what would that be? Who wants to take that?
I will answer on behalf of all the panelists.
Okay.
I would say, even though mRNA vaccines have not been used until now, all the data suggested so far that the authorized mRNA vaccines are as safe as other types of vaccines that have been used for years. and even more effective than some other types of vaccines. So with the emergency use authorization of these vaccines and more vaccines, as we all know now, in clinical trials, I would say we're hopeful that we'll be able to put this pandemic behind us at some time, and probably not in too distant future. However, though, it's going to take a while before we can completely resume our normal life. So we'll need to work together to practice social distancing and continue to wear masks for a while in order for all of us to step out of the dark tunnel altogether.
Thank you, Cynthia. That's a very wise advice. Thank you so much for this. And I think it's time to turn it back to Michael.
All right. Well, Jills, Angela, Catherine, and Cynthia, thank you so much for joining us today. As we prepare for another exciting year of broadcasts in 2021, we invite you to view the hundreds of edited recordings that we have in our library, which is an exclusive ACS member benefit. I would note though that there is one exception to this. If you go to acs.org/webinars, you will notice a COVID-19 tab, and all of our COVID-19 related content is unlocked for the public at this point. We would love to hear what you think about our program, and you can reach out to us through ACS social media channels on Facebook, LinkedIn, and other areas as well. You can also email us at acswebinars@acs.org. We are curious to hear what you thought of today's broadcast, and a short survey will pop up directly after the webinar that you can let us know what you think. In addition, you will also get an invitation email to view the recording from today's presentation. It will be open for a 24-hour period, after which it will be edited and become posted to the ACS website. And that wraps up our program for today. On behalf of all of us at ACS and CAS, thank you for joining us. We hope you enjoy the winter holidays with your families, hopefully safely. And until then, stay safe and stay healthy. So goodbye, everyone.